All right, thanks. All right, so yeah, I'm gonna talk to you about my work on conservation genetics of northern leopard frog in North Dakota. And Craig already talked a little bit about our funding sources. So most of this work is funded by the USGS, Climate and Land Use Research and Development Program, as well as North Dakota Game and Fish, NDWRRI. And I just want to acknowledge some of the people that have helped me out throughout various stages of this project. All right, starting out, I think really big. So what do we talk about when we talk about biodiversity? And it really can be broken down into three basic components. So if you ask your general public person on the street, they'll probably get the first one. That's species diversity. This is your endangered species, your species richness. If you press them for a second one, they might get ecosystem diversity. So save the rainforest, protect coral reefs, kind of preserving biodiversity in these big complex systems. But they probably wouldn't get this third one. That's genetic diversity. And this is really the diversity that's contained within species. And that's what we're going to be focusing on today. So if genetic diversity is so often overlooked as a component of biodiversity, why is it so important? And usually it gets brought up in kind of two main contexts. The first being inbreeding. This is especially dangerous for small populations. So when you have small populations and lose some of the genetic diversity, you kind of lose some of the protective benefits that come from it. And you get these accumulations of mutations that can drive down fitness, causing the population to shrink even more. You lose even more genetic diversity, and you get caught in kind of this extinction vortex. And then the second really big context is adaptive capacity. And this is especially important when we're dealing with environmental change. So you might be familiar with kind of the four ways that an organism can respond to environmental change. It can die, go extinct. It can move somewhere where it can survive. It can exhibit a plastic response, change of behavior, or it can evolve. And in order to exhibit this evolutionary response, you need to have that standing genetic diversity because that's really the raw material that evolution can act on. So that's all well and good, but what can genetic information do for us as conservation practitioners? And there's really a lot of different things we can do, knowledge of genetic diversity. I'll just go run through a few examples that are particularly relevant for this system that I'm going to talk about. And the first is management units. So these are kind of the way we organize our conservation efforts around different populations. How should we treat different groups of wildlife and plants that we want to conserve? So say we have our two ponds of frogs here. We want to know whether or not we should be treating them as like one big system or kind of separate entities. So we can do our genetic survey and see, oh yeah, they're pretty genetically distinct, so we should probably be managing them as separate entities. Another example is in dealing with our small populations. So we can kind of take a look and see if they are suffering from genetic inbreeding. They have low genetic diversity, so we can do our genetic survey and recognize that. But then it can also help out with our genetic rescue efforts. So bringing in new genetic diversity to help out those inbred populations. And one of the dangers in doing this is that we don't want to pick a donor population that's wildly different from our inbred population. We want to try and get a donor that looks somewhat genetically similar, might have some of the same adaptations as our inbred population, but just kind of help bring in some new genetic diversity and help mask those negative mutations. And then finally, you can use genetic information to assess resilience to environmental change. So say we have our red genotype frogs adapted to sunny ponds and our orange genotype frogs adapted to rainy ponds. If there's some climate change and our rainy ponds become sunny ponds, we might lose some of those orange frogs. And then we might want to consider doing some assisted gene flow. So taking some of those red frogs, bringing them over into the orange pond And maybe there's some adaptive genetic variation there that can spread and help that population recover. Alternatively, if we know if there's already some established gene flow between these populations, when we see climate change happen, we might not expect the declines to be so drastic that Orange Pond might be able to recover on its own without assisted gene flow. And instead, our conservation efforts might be better spent trying to preserve that connectivity. Right. So now start talking about kind of the context of this study in particular. So let's start us out with the ecology of North Dakota. So there's really four main ecoregions here in the Red River Valley, on the Lake Agassiz Plains. And as you move west, you hit the northern and northwestern glaciated plains. And then finally, west of the Missouri River, you get to the northwestern Great Plains. And these ecoregions really vary a lot in wetland density. So in the northwestern Great Plains and here in the Red River Valley, we have pretty low wetland density, whereas in the glaciated plains, those really constitute the heart of this region called the Prairie Pothole region. That's one of the most wetland dense places in the world. And then as you go west, there are differences in topography, prairie habitat, precipitation, and land use. So it's really flat here in the east, gets hillier as you go out west. Tall grass prairie here in the east moves to mixed grass and short grass prairie. Wet here in the east, it's drier as you go out west, and a lot more cultivated cropland here, whereas it's shifted to pasture as you go out west. So really a lot of differences across the different areas in North Dakota. And of course, we also experience a lot of environmental change, especially in recent times here. So in terms of climate change, here in eastern North Dakota, you're part of an unprecedented wet period. It's about 1993. And whereas in the western prairie potholes and northern Great Plains, they've experienced more drought cycles. And there's a lot of uncertainty in this precipitation future going forward. So we don't know how long this wet period is going to last, if that decline in between east and west is going to steepen, where that gradient might land on the landscape. Then the other major driving force of environmental change in this area is land use change, really due to agricultural expansion with commodity crops like corn and soybeans. And over 50% of the wetlands have been drained in North Dakota since the 1800s. A lot of habitat loss, a lot of fragmentation, and then also the introduction of agrochemicals like fertilizers and pesticides, which can have further effects to degrade the environment. So now that we've set the stage, it's time to introduce our star player, and that is the northern leopard frog. It's a little bit about their ecology. So as you can see, they're really good at hopping. They have a really high dispersal ability, especially compared to other amphibians in the Great Plains in North Dakota. And they use that really good dispersal ability to get around different seasonal habitats. So during the spring and summer, you're really reliant on these breeding ponds, which are temporary or semi-permanent wetlands without big fish predators where they breed. The tadpoles can develop. Later on in the summer, they really utilize these wet meadows and grasslands as foraging places. And then finally, a really important component here in North Dakota is overwintering sites. And these are deep permanent wetlands or flowing water habitats where leopard frogs can go down and avoid the frost line because they can't freeze solid like some other frog species can. Just a little bit about the conservation status of northern leopard frogs. They are a really widespread species. So you can find them from the Atlantic seaboard all the way to Washington State, British Columbia. But they have faced declines pretty much across their whole range, but especially towards the western edge. And they are fairly common in North Dakota. They're one of the big sources of amphibian biomass. But if you kind of look at some of our neighbors, Saskatchewan and Wyoming, there are some nearby populations that are vulnerable. So kind of broken this down into 4 main chapters with the overarching goal of kind of assessing genetics, the genetic status, and some of the main threats to northern leopard frogs in North Dakota. So in this first chapter, it's going to be kind of just a general overview of the population structure and kind of the population history across the state. In chapter 2, we're going to kind of look more specifically at adaptive genetic variation as well as phenotypic variation. Then for chapters 3 and 4, we're going to zoom into a more local level and look at gene flow and local adaptation due to climate and agriculture. Right, so moving on to chapter 1, so there's kind of 3 main goals here. One's just to characterize statewide population structure. Then we can kind of look at what are the main drivers causing that structure. And finally, kind of look back at the history of how all this structure developed. So here's our map of North Dakota. And to do this chapter, we sampled 41 sample sites all across the state. 30 frogs per site. Each of these frogs was genotyped at 11 microsatellite loci. And this map is color-coded by the major river basins. So what we see when we actually look at the genetic structure is that we can kind of classify frogs into clusters based on these river basins. There's also like a broader pattern in structure, and it is this group over here on the left side of the graph all falls west of the Missouri River, whereas the group on the right all falls east of the Missouri River. That's a big pattern that we see. So now we can start to get at some of these landscape features that might be driving in that structure. And to do this, we're doing this variance partitioning analysis that allows us to kind of break apart how much genetic variation is due to these different landscape variables. And this is going to pop up a few times. So the first pattern we're going to look at is isolation by distance. And this is just things being more genetically distinct because they're farther apart in space. And we see this is a pretty strong pattern. It explains about 60% of our genetic variation. So pretty common to see a strong pattern in amphibians because even leopard frogs can't move that far. Next up, we have our river basins. And after we've already accounted for space just by being in different river basins, that accounts for about 20% more of our genetic variation. And then finally, the Missouri River itself, just by being on one side or the other, that explains about 4% of our genetic variation. So these are all really statistically significant effects on our genetic variation. So that's how kind of structure is organized. But how did it get that way? So in order to do this, we're going to look back in time at the population history. And to do this, we're going to do 2 approaches. First is this DIY ABC. And this is a program we can use to estimate coalescence using our genetic data. So how far back do we have to go before we see a common ancestor between each of our genetic clusters? And also, we can use these species distribution models. So based on the habitats the northern leopard frog can exist in today, where were they likely to exist in climate history going back thousands of years? So our coalescence analysis kind of shows that this big east-west split on both sides of the Missouri River occurred about 11,000 to 18,000 generations ago, which is about 22,000 to 36,000 years. Generation time for leopard frogs is about 2 years. So that's during the last ice age, right around the end of the last glacial maximum. So I do want to point out that during that time, North Dakota is almost entirely covered by the ice sheet. It's only that southwestern portion that remained unglaciated. Then we see that a lot of this population substructure, so the differentiation between the different river basins mostly occurred after that, so about 22,000 to 6,000 years ago. So if we look at the species distribution model, what does it tell us? So we can kind of see that there were two main refugia during the last glacial maximum at the end of the last ice age, one west of the Mississippi River and one east of the Mississippi River. So both of our clusters came from that western refugia. If we kind of play that, you can kind of see that as the glaciers receded, leopard frogs started moving north, and that's when we kind of, here's the mid-Holocene. You can see there's kind of some climate variation going on there. So that's when our substructure occurred between the different river basins. And then we get to their current distribution. So at some point when they were moving north, that's when that big east-west happened. And then probably when they got here during those climate fluctuations during the Holocene, we see that substructure develop. All right, so moving on to chapter 2. So this is kind of related, but instead of looking at neutral genetic structure, we're going to look more specifically at possibly adaptive genetic variation as well as phenotypic variation. And then we can also kind of compare and contrast patterns and what might be driving those patterns in genetic variation versus phenotypic variation. So we collected phenotype and genotype data from these 11 sample sites, representing each of the 4 major ecoregions, and they have really different land cover terrain. So grassland versus grassland and pasture versus cropland, very different wetland densities. So remember, those wetland densities are going to be highest towards the middle of the state. And then we also included a couple forested sites. So there's this Turtle Mountain. It is a lower level ecoregion, but it has really different land cover compared to the other areas. We thought there might be some interesting genotypic and phenotypic variation up there. And then we also captured a lot of climate variation. So this first map at the top kind of shows precipitation and temperature gradient, where the west is generally drier, warmer than the east. Then we also have some seasonal variation from the south to the north. And this genetic data is based on RADSEQ data. So this is going to be how we collected genetic data for the next 2 chapters going forward as well. But we collected genetic samples from 20 frogs per site. They're genotyped using BestRAD. What this approach does, it takes little snapshots of random spots across the whole genome. We're looking for these single nucleotide polymorphisms, or SNPs. And what these are, just a single base pair change, one single point mutation. And a frog could be heterozygous, this spot, or either of the 2 homozygotes. So we have 3 possibilities for genotype, but instead of having just 11 microsatellite loci, now we have about 3,000 of these SNPs in each of our data sets going forward. So what do we see in terms of genetic structure among our 11 sample sites? Well, again, we see this really strong east-west split. We have our blue group west of the Missouri River, and then our red group east of the Missouri River. And then looking at substructure, what we see is, a little bit of uniqueness up here in Turtle Mountain. So right up there. And then we kind of see that the western populations might have a little bit more substructure than our eastern populations, which tend to be more homogeneous. And then our phenotypic variables. So we measured 17 morphological traits, mostly looking at landmarks in the head, as well as some measures of limb length, as well as a few color variables. So leopard frogs, famous for their spots, did spot counts, as well as color morphs. So there's 2 main color morphs, brown frogs and the green frogs. And then there's also this really cool regional pattern morph called the Candy-Gohi morph, where you have this dark black vermiculation in between the spots. There's only a few of these frogs in the data set. They don't have a huge impact, but it's really neat. So what do we see when we look at phenotypic variation? We see that a lot of it's contained within sample sites. So there's not a whole lot you can use to distinguish between different sample sites. When we look at a broader level, we see a little bit of a difference between that western cluster and the eastern cluster. That western cluster tends to have proportionally longer legs, but it's not a hard, clear difference. As you can see, there's a lot of overlap between those. Just on average, it is significantly larger. Okay, so now we can start to look at whether or not the environment or some other landscape processes are explaining patterns we see in our genotypic and phenotypic variation. So we're going to look at a couple different patterns. We're going to look at isolation by distance again. We'll also look at isolation by environment. So instead of how far apart things are, how different are the environments that they're found in? Does that explain how genetically or phenotypically different they are? And we'll be using those climate and land cover variables that I talked about at the beginning of this chapter. And then finally, demographic history. So this is which genetic cluster do they belong to? Which of those river basins, basically. And again, we're looking at variance partitioning. So when we look at genetic variation, what are the importance of each of those patterns that we see? We see that they're all pretty important. So our isolation by distance explains about 7%. Our isolation by environment, almost 20%. Your demographic history, a little over 10%. But when we look at phenotypic variation, we see kind of a different result. And that's our isolation by distance and isolation by environment don't really explain a significant amount of our phenotypic variation. It's really only demographic history that explains a good amount of our phenotypes. So that kind of suggests ancestry might be the most important component in phenotypic differences, but there's a lot of possible plasticity there too. All right, so moving on to chapter 3. This is, you might notice the background is from the classic arcade game Frogger. This is kind of the Frogger chapter. So this is assessing connectivity between different sample sites. How easy is it for frogs to move? So we'll look at connectivity. What are the major landscape factors affecting that connectivity? So for the next 2 chapters, we're going to be zooming in to a more local level. So you can kind of see that we're back at our river basin map here. And the next 2 chapters are going to focus on one of these river basins. So this is the James River, Lake Oki river basin in the purple. We sampled 28 sample sites, 28 different wetlands across this area. And you might notice that they're broken up into a couple categories, all based on how much, based on the type of land cover in the area around each of these sample sites. So we have 7 sites with the most natural land cover, mainly surrounded by grassland and wetlands. We have 7 sample sites with the most agricultural land cover. So these are sample sites surrounded by a lot of cropland. Then we have 14 of these paired sites. And these are selected to have the biggest, be close to each other and have the biggest differential. So one of these sites is going to have a lot of agricultural land. One of these sites is going to have a lot of natural land. And these will be really helpful in being able to distinguish between effects that are due to agriculture versus just space. I'll start you out with some good news. We don't see any genetic fragmentation due to agriculture in these sites. You can look at a couple different measures of genetic diversity. There's just one breeding coefficient. So this can vary from -1 to 1. If you look at our estimates, most of those error bars cross 0. So that's good. 0 is no inbreeding. And even The ones that are significantly different from zero are pretty low, not enough to cause a concern. And in our high agricultural areas, they're just as likely to have negative inbreeding values, which kind of indicates possible outbreeding. So really no sign that these frogs have lost any genetic diversity due to agriculture. And now we want to start assessing how the landscape might be affecting gene flow. So we need to know what our landscape looks like. So we kind of broke our landscape into landscape variables in a few major categories. First off, dealing with land use. So we have our amount of cropland and grassland and urban areas, open water, all those different land classes. And we also specifically looked at the frequency that a few of the main crops were planted. So we looked at corn, soybeans, and wheat. And then our next category is desiccation risk. So what is the likelihood that a frog is going to dry out at some point when it's moving between these different habitats? So up here we have on the left distance to a permanent wetland. So these would be those overwintering sites, heat load, which is the amount of solar radiation a surface might get, topographic wetness, which is where moisture will tend to accumulate on the landscape, and the number of rainy days area gets during the spring and summer, because we know that frogs like to move during rain events. Then we also looked at topography, so just how much elevation change, and then major roadways, so highways and freeways. And to do this analysis, we need to simulate gene flow, and we use this program called CircuitScape. And you can really, the way this program works, that it treats gene flow like electricity. So you can kind of think of our landscape as a circuit board. The landscape variables kind of supply different resistance values in that circuit board. And then our gene flow is going to follow the routes of least resistance. And then we can compare resistance distance. So how long does it take for an electron or a frog to move through our circuit board to our different sample sites, the genetic data, and see which resistance surface is the best job at explaining our genetic differences. So just to kind of help you visualize that, here's our land class data. Say we want to give grasslands a low resistance value, make it easy for frogs to move through. We can give cropland a moderate resistance value, make it a little bit harder. We can give like open water, urban areas, really high resistance value, make those act like a strong barrier. We can test out different combinations of these resistance values and see which matches our genetic data best. And then we can also combine different resistance surfaces. So we can take our land use and add it to our roughness resistant surface, kind of get these more complex surfaces, see if they do a better job of explaining our genetic impact. So once we do all these different combinations, what do we get? I kind of spoiled it. It's land use and topography that best explain our patterns of genetic diversity. What we see is that grassland and other natural habitat types tend to have the lowest resistance values. Urban, low intensity urban areas like suburbs and pasture land have moderate resistance values. So still pretty easy for frogs to move through. Cultivated croplands are kind of a permeable barrier, so it's not super easy for them to go, but they can still kind of move through it. And then areas like open water, high intensity urban areas, be like downtown of Jamestown and Bismarck. And then curiously enough, wetlands have really high resistance values. And the wetlands thing, sometimes you get this weird effect where the core habitat area ends up having high resistance values. Kind of think about it from a frog's perspective. You can't live in a grassland. But once you come across a wetland, hey, that might be a good place to stop. And then roughness, a pretty simple relationship. You have flat land with low resistance, so it's easy for frogs to move in flat areas versus a little bit harder for them to move through hilly areas. All right, and on to the last chapter. So now we're going to be looking at local adaptation. So we'll look at, again, how the environment might be structuring genetic diversity on these finer scales, and then try and find if there are any specific genes that might be under selection due to climate and agriculture. So looking at population structure in this area, we see, well, there's kind of 4 main clusters. So we have this blue population kind of around the Jamestown area, red, this red population up on the northern part of the Missouri Plateau. The northwestern part of our study area, we have this green Sykeston population. And finally, we have these 2 populations more along the Missouri River down by Bismarck. So we want to kind of look at what role the environment might be playing. We'll look again at those different patterns to try and assess how they're affecting our genetic variation. So isolation by distance again, and then isolation by environment. This time we'll break down isolation by environment into 2 kind of subcategories, one for climate, one for agriculture. So for our climate variation, we ran a principal components analysis and made kind of these composite variables. I'm just going to pull out kind of the main big one here. And these are all different trends in temperature and precipitation. But kind of the big one here is this principal component 1 that explains about 45% of the climate variation in our populations. And it's mainly this trend from hotter and drier summers in the southwestern part of our study area to cooler and wetter summers in the northeast. And for agricultural variation, we looked at different estimates of pesticide exposure. So we looked at 4 different pesticides. One of these is atrazine. So if you're familiar with amphibians and atrazine, there's kind of a notorious relationship there. Atrazine is an endocrine disruptor in amphibians and can cause intersex problems and feminization of male frogs. We also saw that 2 of our other most commonly applied pesticides, glyphosate and acetochlor, have almost the same spatial pattern as atrazine, so we can't actually tell the effects of those apart from atrazine. We also did look at 2,4-D, which is a different herbicide. It's mainly applied to wheat here, whereas the other ones are mainly applied to corn and soybeans, so it had a different kind of spatial distribution. We actually can tell it apart from our atrazine. And then we also looked at nitrates based on this USGS groundwater data. So looking at our variance partitioning analysis, we see that isolation by distance explains about 3% of our genetic variation, agriculture about 3.8%, climate almost 6%. And all of these are really highly significant, so it may not sound like much, but these are all really highly significant patterns that we see. So now that we know that there is some of this genetic diversity correlated with these environmental variables, we can look for certain genes. And to do that, we need to find outlier SNPs. So we use these outlier detection tests. There's a whole laundry list of different ones you can pick from. They all do basically the same thing, that they're looking for SNPs that don't match our patterns of neutral population structure. And some of them will even look for SNPs that are specifically correlated with certain environmental variables. So I picked out 4 of these methods. We have 2 versions of redundancy analysis, PC Adapt and BAM2. All you really need to know about these is that the statistical stuff under the hood is a little bit different. But what we end up doing is that we look for overlap In between these four methods, what we find is that there are 57 outliers detected by at least two of these methods. So these are candidate loci under selection. So one of the first things we can do with these candidate loci is see which environmental variables they're associated with. So when we do that, see that most of them are related to climate. So 82% are driven by climate, and it's mostly that PC1, so the spring and summer conditions in east versus west. So here's an example of one of our sites. You can see this gray allele is more common in the dry, hot areas, whereas the other major allele in black is more common in the wet areas. But we also see a pretty good number, 25% of our SNPs are associated with agricultural variable. That is almost always going to be either 2,4-D or atrazine. So we know what environmental variables these SNPs are related to, but can we figure out what these SNPs are actually doing? And to do that, we need to align them with certain genes. So we align them to the European common frog genome. This is the closest relative of the leopard frog with an annotated genome. And we see that 29 of our SNPs actually occur in an intron, so pretty good. It's in a gene, at least. It's not going to be the mutation causing the fitness difference, but it might be linked to something. One of them occurs in an exon, the actual protein coding region of the gene. 25 are between genes, so they still might be linked, but we don't necessarily know to which gene. And 2 just had no match at all to the European common frog genome. And when we look at the function of the genes that are associated with certain SNPs, we see a wide variety of functions. 2 of the main ones that pop out are DNA replication and repair, and ion transport. So these are pretty broad categories. We can't narrow down like a certain pathway that might be under selection, but it gives us a starting point to maybe look at some possible adaptations. And about those starting points, so I do want to highlight just 2 genes that were part of, that were singled out in relation to pesticides that also have some effects of pesticides shown in other animals. So we found this TET3 gene, which is related to epigenetic changes in Japanese Medaka. So that's the little rice fish on top, and they're exposed to glyphosate. And this was correlated with our atrazine variable, which, if you remember, also includes glyphosate and acetochlor. And then this aquaporin AQP4 gene is related to water management in the cells, mice. It's affected by atrazine as well. All right, so just to summarize some of our main takeaways, what we see is that there's strong population structure, the Missouri River acting as a barrier, and this population structure largely occurred due to dispersal northward out of those climate refugia after the last ice age. And we see some phenotypic differences, definitely really strong genotypic differences. From chapters 3, saw that there's pretty low fragmentation and high gene flow throughout our leopard frog populations, despite agriculture, and our cropland is kind of a permeable barrier, whereas flat grassland is really great in terms of maintaining connectivity between our leopard frog populations. And then finally, see that our leopard frog populations are probably appear to be locally adapted here. So we see strong effects of isolation by environment, pretty much in all the spatial scales that we looked at, as well as we found some of these outlier loci associated with climate and agriculture. So hopefully you're going to come away with this And notice that environmental change poses risks even to common species like the northern leopard frog. And it's really important for us to keep these common species common. Common species abundance can have just as much an effect on ecosystem function as the presence of endangered species. And we don't really have a good replacement for leopard frogs in North Dakota for what they do. And that this genetic data can be really valuable in terms of conservation planning. So from our big takeaways, might say that, hey, we should be organizing these leopard frogs into separate management units, east and west of the Missouri River. If we want to maintain connectivity, both to maintain that high genetic diversity and to help spread adaptive alleles as the environment changes, we really need to preserve this grassland habitat. And then finally, in terms of local adaptation, we have some interesting questions surrounding this. So what types of adaptations are going to be helpful for climate in the future, as well as, just because these frogs are like they're adapted to pesticides, are those adaptations really going to help them out enough in order to keep those populations viable? So that's about all I have. So I'm happy to take any questions. Yeah. Thanks.
So you said they would have longer legs in the west.
Yeah. Yeah. So kind of one of our hypotheses, a priori hypothesis going into that limb length might be longer in the west because the wetland density is lower. So it's going to be a little bit further distance on average to get from wetland to wetland, possibly. So that's one hypothesis. But there's a lot of plasticity. So it could also limb length be affected by temperature and food availability during the metamorphosis period. So yeah. And I don't know in the northern leopard frog what kind of the spatial distribution is, but kind of these morphological traits are really common to separate species of, related to northern leopard frogs. So it's part of a large species complex, and there are some morphological differences between those species. Yeah. Thanks.
Flip back to your chapter 4, we have the little frequency map. Yeah, that's great. So one thing that's struck me, it seemed like those two populations to the far east are a little bit funky in terms of like their real proportions. Like they have more two than the rest. So I was wondering if you could like speak maybe in terms of like what you saw out of those like field sites or things like that that might be kind of driving the extra allele twos in those two potential parties.
Yeah. So this is Turtle Lake, and these guys. Yeah. I don't really know. These are two of the really high agricultural sites. So this is actually kind of like right on the highway on your way to Jamestown. Yeah, I wouldn't really expect it to be like that much different. There's not like a huge sample size once you filter out a lot of the data. So a lot of these are based on like 8 frogs per sample site. So there's a little bit of randomness in there too. So I don't know either whether have that warm allele.
There is one comment your artwork. art.
Yep. There's the northern leopard frog that Dave has hanging in his house now. Right. Well, if that's it, I just have one more little thing. And I just want to give a special thanks to everybody who's kind of helped me out along the way. So an extra special thanks to Craig and Dave for setting up this project, letting me loose on it. Special thanks to my family, who's supported me along this whole way, as well as to my lab mates and the D&D group, who I think all made it into the Zoom, as well as Brian, who's here. So thanks everybody for showing up.
We will keep this channel and think of how that. Did a nice job, Chris.
Thanks, Bridger.
Yep.